COVID has affected different aspects of science and engineering and medicine, as well as our connection to society. We'll have some of the leaders around the world of both scientific societies and national academies to talk with us. So stay tuned for that in November. And now it's my very great pleasure to introduce my colleague and friend, André Nel. So André is a distinguished professor of medicine and is the founder and chief of the Division of Nanomedicine at UCLA. He's the director of the University of California Center for Environmental Implications of Nanotechnology. As you've already heard, he's one of our editors at ACS Nano, and he bridges science, engineering, and clinical medicine. And so we're delighted to have him with us today, talking about very recent advances in the development of COVID vaccines through the use of nanomaterials. And André, the stage is yours. Welcome.
Good. Thank you, Paul. I'm going to switch to my screen share view. Just a second. Okay. Finally did open, and then I just want to set my pointer options. Well, thank you very much for that introduction, Paul, and again, thank you to Paula for a most enlightening talk. What I'm going to do today is to talk to you about COVID vaccination, drawing on major progress in nanomedicine for infectious disease and cancer. As you know, nanomedicine has introduced an engineered approach to medicine with major impact on therapeutics, diagnostics, imaging, and prevention. Whereas in the earlier years, a lot of the advances have been in therapeutics for cancer, infectious disease. We're now also seeing the emergence of more and more technology applications for cardiovascular disease, anti-inflammatory, and regenerative. And Paula has given us an excellent outline on the left-hand side here about regenerative applications. What I want to talk about today is the impact on immunology for vaccine development, and then also that where a lot of the knowledge have been derived from cancer immunotherapy. And I hope that I will be able to convince you that what we're doing in this phase of nanotechnology is to make use of nanoparticles to mimic viral interactions with the immune system. So nanoparticles in the immune system, nanoparticles can be engineered to either avoid interactions or to specifically drive interactions with the immune system. And you can do it for immune stimulation, for desirable outcomes such as vaccine development, and then there's also undesirable outcomes such as inflammation. You can also suppress the immune system in a desirable fashion, and that is the technology that we've developed for autoimmune disease that I will not be talking about today. And then, of course, undesirable outcomes of immune suppression then would be that you would lower the body's defense to infectious disease agents. Important is that in the interface of a nanoparticle with the immune system, what we call the nanobio interface, a lot of design characteristics of nanoparticles such as shape, size, charge, etc., the ability to deliver, can be designed to promote the direction in which the immune system will go. For example, one of the features that nanoparticle can impart on the immune system is known as danger signals of the aluminum oxyhydroxide nanorod, which is made to replace alum as a vaccine agent, brings in a particle where a high aspect ratio is used as a danger signal to drive specific events in the dendritic cells in the immune system to promote a skewing of the immune response that's known as a Th1 immune response. I'll come back to that later on. Don't worry if you don't understand at the moment what that is all about. Now, in the consideration then for COVID vaccine, the important considerations in this pandemic is to select an antigen for antibodies and T-cell responses. And this can either be a whole organism, a subunit, or pieces of the virus epitopes. Another important consideration is the presence of an adjuvant. An adjuvant kickstarts the immune system and imparts those danger signals that I've just spoken about to drive the presentation of the antigen that is chosen for presentation to the immune system in different formats. And then also of key consideration is that the technology be scalable, and the cost is an important factor in the current pandemic. So just as a brief introduction, how do we make the vaccine simulate natural COVID viral immunity? Well, if you take the COVID nanoparticle, the virus enters via a spike protein, binds to an ACE2 receptor into epithelial cells in the lung and gastrointestinal tract, where the virus is released intracellularly, releases the viral RNA that instructs viral protein synthesis that assembles into new viruses, which buds out of the cell, can infect new body tissues, as well as the cells of the immune system. In this particular instance, where the RNA is translated into proteins that are presented via major histocompatibility complexes, it drives the T helper and B cell responses to produce anti-coronaviral antibodies, which have the important function of preventing the virus from binding or tagging it for destruction. The other side also drives the development of cytotoxic T cells, which have the important function to destroy the already infected cells and to provide viral clearance. An important additional component is that you ultimately also need to have memory of the system through the development of long-lived memory B and T cells to provide durable immunity. So the World Health Organization lists about 170 different vaccines that are currently in development, with about between 30 to 40 in clinical trials. And what is significant is that these vaccines make use of contemporary vaccine technology. I'll explain this in a moment. Live-attenuated viruses, inactivated viruses, or what we know as viral vectored vaccines. What I'm going to focus on today is going to be on the next generation nano-abled vaccines that are aimed at delivery of DNA, RNA constructs, as well as proteins and peptides that can be used for immune generation. So let me just say a brief word about the non-nanotechnology vaccines, very important. The live-attenuated and inactivated viral vaccines, where the whole pathogen is used, either weakened or inactivated. An example of a weakened virus for the current epidemic is the codagenic intranasal vaccine for a stimulation of systemic immune response through mucosal surface. These vaccines tend to be highly potent for humoral and cellular immunology. Single dose administration, which is a huge advantage in the future vaccination efforts that's upcoming. This is going to be a big factor as to whether you deliver 1 or 2 doses. Theoretical safety concerns that luckily have not shown up so far is that weakened viruses can revert back to the pathogenic form, can be reactivated in immune deficient people, or can recombine with circulating wild type viruses. And another potential downside is that these vaccines needs to become refrigerated for distribution, so that somewhat limits the applicability. Several COVID-19 vaccines are inactivated. One example here is, for instance, the Sinovac vaccine that's being developed in China. The inactivated vaccine is possibly safer, and but potentially less immunogenic than the weakened virus. This requires multiple dose administration and also requires an adjuvant. I will review that in a little bit. The Sinovac vaccine uses alum. Recent publications from Sinovac shows the development of an inactivated vaccine that is proceeding, as far as the scientific aspects are concerned, very successfully, with clinical trials soon starting. Viral vectored vaccines make use of live viruses in which some genes have been inactivated to prevent replication. So this is it's not a coronavirus, but it contains coronavirus genes which are delivered, and that becomes the antigen. Examples of such viruses is the non-replicating adenoviral vectors, for instance, the type 5 adenoviral vectors, and the vaccines currently examples are CanSino vaccine, Beijing Institute, and the Canadian Centre for Vaccinology. In the UK, a vaccine that's developed uses the chimpanzee adenoviral vectors, and that's the vaccine from Oxford, AstraZeneca. The advantages of the viral vectored vaccines is broad tissue tropism, inherent adjuvant properties, and scalability. Potential disadvantages is pre-existing immunity to the adenoviruses that may dampen the efficacy, and then also the size of the gene that you put into the virus, limited packaging capacity for long nucleic acid sequences. Recent publications by Dr. Yu et al. Describes the safety, tolerability, and immunogenicity of the CanSino vaccine, published in Lancet. Dr. Folagati recently published about the Oxford vaccine that in their preclinical studies had acceptable safety profile and boosting of spike protein immunity. I should say that last week the trial was temporarily stopped because of one severe adverse event. What I want to talk about today is the next generation, or nano-abled vaccines. These vaccines can either be delivered by nanoparticles, that could be lipid polymer or liposome nanoparticles, or by a new generation of particles known as viral-like particles that are constructed by the assembly of viral proteins. These vaccines deliver also new classes of vaccines, not just proteins. DNA, RNA, and then can also be made for delivery of subunit vaccines and peptide-based vaccines. The advantage of this technology is that there's viral mimicry by the nanoparticles. Looks just like the virus in interacting with the immune system. You can entrap and protect bioactive molecules such as the nucleic acids. Antigen-presenting cells prefer particulate antigens. You can do multi-cargo loading, antigens, nucleic acids, adjuvants. You have control over biodegradability, control over biodistribution and lymph node targeting. And you can also exert control over the intracellular release to allow the major histocompatibility complex to present to the immune system. And then, very important, scalable manufacturing. And again, these are representative publications. I will just say something about the publications that I'm talking about today. Most of what I will show you today have been published in the last 3 months, or some publications still have to come out. So this shows how active the field is. So nucleic acid vaccines is a new concept, not yet FDA approved. You can either deliver DNA, which also then requires technical intervention with electroporation, that makes it somewhat more complicated, or RNA, in which you can use nanoparticles or viral-like particles. The advantages: speed, scalability, and reduced cost. Very important for the pandemic. All antigens are processed, enters the immune system looking like a virus, so there's good viral simulation. You can generate antibody, CD4 helper, and CD8 cytotoxic T cell responses, and the nucleic acid has important intrinsic adjuvant activity. Examples of DNA vaccines currently: Inovio, Entos, Zydus. Examples of RNA COVID viruses: Moderna, Acutus, Arcturus, and technology from Pfizer and Pfizer. The messenger RNA or RNA delivery have some advantages over DNA in that the RNA only has to enter the cell. So there's only cytosol delivery, no genome integration such as for DNA, not labeled as a gene therapy. And the RNA is transiently expressed, quickly degraded. There's faster synthesis and purification and a lower cost because no cells or proteins are required. And also the RNA can be delivered as just the RNA strand from the virus, and that's called a non-amplifying vaccine, or the RNA can be delivered with self-amplifying constructs where both the viral replication machinery and the antigen contributes to synthesis of increased amount of RNA-related protein and protein expression. A lot of ingenious design has gone then into the developing of the nanoparticles for the nucleic acid delivery, and I'm going to be briefly talking to you about it in three different contexts. The one is, how do you choose the nucleic acid design? Second is, how do you package the nucleic acid? Describing here the development of lipid nanoparticles, which makes use of unique design components to package and to deliver. And then another aspect that I'll briefly describe is if these particles enter, like the intact coronavirus, you need to get that RNA out of the endosomal compartment. And so there has to be release of the RNA. So how did we get to choose the antigens? Important lessons came from MERS and SARS and its spike protein. SARS, of course, is the severe acute respiratory syndrome coronavirus, emerged in 2003 at a high case fatality rate of 14 to 15%. Not as deadly as the MERS, the Middle Eastern respiratory syndrome coronavirus, which emerged in 2012 and had a case fatality rate of 36%. Important lessons is that the spike protein mediates viral uptake and membrane fusion. The S protein and its domain was earmarked as targets for making neutralizing antibodies. So making neutralizing antibodies was a prime objective of the vaccine efforts, as they currently are for COVID-2. We'll discuss the relative benefits as well as potential downfalls of that. Importantly, the antigen or the S protein was smartly designed to assume a certain conformational integration of the virus to enhance immunogenicity. So let me just focus on that, how that came about. So on the left-hand side here, show you MERS-SARS virus, also mimicked now by COVID-2, in which the spike protein assumes a lot of the attention. But what I also want to point out to you that the spike protein is by no means the only immunogenic part of the virus. The nucleocapsid, the membrane, and envelope proteins are also important. What is important from the perspective of the spike protein is where the virus is released. The spike protein attaches to new cells where proteolytic cleavage of the viral surface allows the virus to be taken up. When the gene was cloned for the S protein at viral entry, it was revealed that it consists of an S2 subunit and a trimeric S1 subunit. It is the trimeric S1 subunit that binds to the ACE2 receptor that's responsible for viral uptake. Further work, when the S2 crystal structure was revealed, was it is a specific binding domain in S1 that interacts with the ACE2 receptor, and that's also called the receptor binding domain. The receptor binding domain, where it sits on the S2 stalk, can either lie down or stand up. And it is the stand-up conformation that we want to achieve for vaccination. Why? Well, The reason is you need to have that RDB stand up after cleavage by a protease. That allows the RDB to assume a new conformation, stand up, bind, and interact with the ACE2 receptor. That interaction with the ACE2 receptor occurs stepwise, where first one RDB subunit stands up, then a second, then a third, and ultimately the virus is then drawn into the cell, and the S2 subunit is drawn out into a peptide. Now, why this is important from an immunogenic perspective is not only does the RDB in the up position provide binding interaction, but all the important epitopes that is instructed in the immune system and to which the antibodies provide protection of the virus sits in this hinge region under the RDB. And if it's in a down position, that's a viral protection mechanism, the epitopes are hidden. So a lot of design has gone into the mutational stabilization in order to provide a vaccine in which the S1 protein is made into a stand-up formation. And I'm not going to go through all the details, but that involved making mutations in that furin-sensitive cleavage site, plus 2 proline residues that sit in an heptamer repeat in the S2 that pushes the RDB out. And you can see from this negative stain EM that for the mutated MERS, the 2 proline mutations and the SARS-2 proline mutations, you have the trimeric presentation only, whereas for the wild-type vaccine, you have the pre- and the post-fusion complex. Okay, so now you've seen how the nucleic acid is designed. Once the nucleic acid sequence is determined, important design features that have gone into the nucleic acid is to design in 5' and 3' untranslated regions that are important for the RNA stability, and that is also comes together with chemically modification of uridine in order to stabilize that RNA. And I've already explained before that you could have it as a conventional RNA. Examples include Moderna and Pfizer vaccines, or you could have a slightly more complicated self-amplifying RNA replicon, such as in the Acutus and the Arcturus vaccines. Okay, now you have the nucleic acid. How are you going to package it? Well, important design have come from the use of lipid nanoparticles. The lipid nanoparticle that we know best is the liposome, in which the structural and helper phospholipids and cholesterol is designed where there's cylindrical shapes of these lipids and they package into lipid bilayers. In very clever design for the viral RNA delivery has come from the inclusion of cationic ionizable lipids, of which there are a number, but the basic idea is that you have an amine head group, you have a linker, and you have hydrocarbons. And these lipids assemble into cone-shaped structures that don't want to make a lipid bilayer. They tend to want to make penny rolls. And this is also known as a hexagonal phase. And in order to synthesize those lipids, also another spin-off in the nano world is the ability to do high throughput synthesis through microfluidic mixing, where one inlet allows in the RNA, the other inlet allows in an ethanol solution, which is then stirred and very effectively mixed with various different mixer types, for instance, a herringbone mixer. The cationic lipid binds the negatively charged RNA, and its hydrophobic tail is inverted, so you get these inverted micelles. Then comes in the other structural lipids, such as the polyethylene glycol lipid. As polarity increases with mixing, the polar lipids ultimately assemble onto the surface of those micelles and coalesce into electron-dense cores that are surrounded by a lipid monolayer. With those electron-dense cores in place, we now have a lipid nanoparticle where it enters an endosomal compartment as the cationic lipids on the outside. They don't want to sit in a lipid bilayer, and they begin to interact with the endosomal lipid membrane, leading ultimately the viral lipids blending with the endosomal lipids and allowing the RNA to escape. And in the process, the cationic charge on those lipids also turn on the proton pump, which fluxes in water, and this helps to push out this RNA so that you get smooth delivery without damaging of the endosomal compartment in the cell. Finally, the feature then of bringing in the nucleic acid and the cationic lipids is the important adjuvant effects that follow from that. So when you inject the vaccine in the muscle, here in the muscle site, you can see there's inflammation. Well, that inflammation is derived from the fact that the nucleic acids and the cationic lipids provide immune danger signals, similar to the immune danger signals that I described earlier on. For instance, a series of toll-like receptors and other receptors. Forget about all the names. These act as sensors for chemokine and cytokine production. The cytokine and chemokines is responsible for this inflammation and recruitment of antigen-presenting cells. The same danger signals, once the antigen-presenting cell comes in, is responsible for maturation of the dendritic cell from immature to mature format, in which there is then MHC expression and any antigen that is expressed being presented by the MHC complex. The nucleic acids also drive these dendritic cells to favor a specific type of immune response called a Th1 immune response. I'll explain in a moment what that is. The nanoparticle design is also important. Nanoparticles of less than 150 nanometer is preferred in order to enter the lymphatic place. Okay, so now you understand something about the new generation of vaccines. I'll be saying something about the subunit vaccines in a moment. But this is an important enterprise in order to drive herd immunity in the human population. And Graham recently making the comment that if we leave immunity solely to natural infection by the virus, even at a 1% mortality rate, 40 million people could succumb to COVID-19 globally. So the efforts are very important. Okay, so what have we learned then from MERS and SARS-CoV-1 vaccines in using the S protein to be the focus of vaccine development? Well, in these vaccines, animal vaccination studies showed very effective development of neutralizing antibody to this S protein or its receptor binding unit that blocks viral entry and provides a protective immunity. However, at the time when in these animal models they were challenged with CoV-1 or MERS, it revealed the emergence of immune side effects that was not previously expected. And so it's very important in the choice of the vaccine components and adjuvants to design them to avoid these adverse effects. What are these adverse effects? Well, I'm briefly going to just say there are two classes. One is adverse effects that's antibody-mediated, so-called antibody-mediated enhancement. Another one is a cellular side effect that leads to damage to the lung. The antibody-mediated side effect is where instead of making neutralizing antibodies, non-neutralizing antibodies bind the virus and increases the rate of cellular infection. And this is a particularly IgG1 antibody class. A second adverse effect is where the antibodies take the virus into an endosome, where you have all those receptors that provide danger signals, and this leads to serious exacerbation of inflammation. The second type of side effect is mediated by T cells, which call into the lung eosinophils that damage the lung. This was first detailed when respiratory syncytial virus was developed in humans and where there was mortality in the kids at the time of RSV infection. Similarly, several MERS and CoV-1 vaccines, and I show here an S protein vaccine or an inactivated viral vaccine, had this post-vaccine enhancement of eosinophilic lung damage. eosinophilic lung damage is shown in the right-hand side. This happened across multiple animal models. And importantly, in those studies, it was revealed that if you use the S protein, there are fewer of these cases. And if you use a Th1 skewing adjuvant, you get fewer side effects. Okay, so let me explain briefly what that is. What is shown here is that the skewing that favors T helper 2 versus T helper 1 is what is responsible for a lot of these side effects. Th1 skewing is what you want, and that is where the vaccine induces a population of Th1 CD4 T cells and cytotoxic T cells in an environment where there's interferon gamma production and where the B cells switch to making an antibody type known as IgG2A. The opposite end is where you have T helper 2 development. This is where what you don't want. In this case, a T helper 2 lymphocyte is instructed in a cytokine environment where there's IL-4 and IL-5. The IL-5 is responsible for pulling in the eosinophils. Here, the B cells make an IgG1 antibody. The possible explanations of why this have developed is the choice of the viral components, that nucleocapsid protein or whole viral vaccines are more prone than the receptor binding domain. The use of Th2 adjuvants, alum, also provides potentially provide Th2 skewing. Aging in the immune system is also an important factor. So the solution to preventing unwanted Th2 skewing is to increase Th1 skewing by the nucleic acid backbone, by inclusion of the S protein or RDB compared to other viral components, or to provide Th1 skewing adjuvants. I'm not going to go through the whole list, but this could include inulin polysaccharides, so-called CpG oligos, the aluminum oxyhydroxide nanorods that I've shown earlier on, as well as a series of lipid-based agonists that drive toll receptors. So a brief check then on one of these vaccines, the vaccine for delivering mRNA 1273 for COVID-2. How well did it do? Well, in animal studies, induced very effective binding antibodies to those domains. These antibodies were of a neutralizing type, which very effectively suppressed the viral load. So vaccination was successful. In looking at the balance between IgG1, IgG2A, or the cytokines from the spleen, it was shown that there was a predominant Th1 skewing of the immune response. So that was good. That's shown by the fact that interferon gamma dominated over IL-4. Interesting, in the same studies, the people also had a control where they just put the S protein and the alum, and look what happened. Made more IgG1, and so the ratio dropped, and it also, in the cytokines, there was a skewing of IL-4 over interferon gamma. What happened when this vaccine was used in trials in humans? Well, in the phase 2 clinical trials, it was shown that the vaccine administered in the deltoid muscle on days 1 and 29 led to rapid seroconversion, antibodies being produced by day 15 with a very good titer of neutralizing antibodies made after the second vaccination. The antibodies was directed to the mutated S protein, correlated well with IgG titers to the RDB domain. The antibody titers correlated well with neutralization of viral antibodies, so there was protection. And typing up of the immune response by using peripheral blood lymphocytes, the response was Th1 skewed. And then in addition, most of the vaccination could happen with very mild or at the most moderate systemic side effects. Okay, now we have the vaccine. The vaccine seemed to be making protective antibodies. How does that correlate to what we now know about the COVID pandemic so far? For humoral antibody responses in COVID-2 patients confirm the immune dominance of RDB and viral S protein in protective immunity. Most symptomatic COVID patients make anti-RDB antibodies by day 9. There's a good sensitivity and specificity. There's a good correlation of these antibodies in the RDB titers to neutralizing antibody titers. But 33% of patients had a poor neutralizing antibody response, even although they made anti-RDB. We learned that anti-RDB, IgG, is an important marker in serum. We have a convalescent plasma serum ongoing at UCLA and in the United States, and this is a promising correlation with protection. An ELISA assay for RDB is also a potential useful tool for identifying plasma donors. Antibody responses also include IgM and IgA, but they are shorter lived. But I want to caution that IgA is mostly made in mucosal surfaces, so IgA in the mucosal surface may not be short-lived, but may play an important role. So an important synthesis from what we've seen in the natural infection that we now have to assemble in vaccine development is that antibody responses is not detectable in all patients, especially people with less severe infections. And that comes with a question is, are they unprotected? Do T cells make a difference? I also want to say something then about the duration of the neutralizing antibody responses. Antibodies against COVID-1 and MERS lasted for 2 to 3 years. Antibodies to seasonal cold coronaviruses, which is also around, are short-term responses allowing reinfections to occur. In a recent study from UCLA, early indications were that for COVID-2, the RDB vaccination response has a half-life for the antibodies of 36 days, saying that perhaps it's not going to be long-lasting, but more data is required. The current expectation, and I certainly hope this turns out correct, is that vaccines that generate neutralizing antibodies provide a viable option for COVID-2 herd immunity. I also want you to recall what I said earlier on. While neutralizing antibodies block viral entry, cytotoxic CD8 T cells are required to eliminate infected cells and viral clearance. T cells also confer immune memory, and immune memory against conserved viral antigens other than the S protein and the RDB. So other proteins may also be important. And we also learned that the best vaccines, the most durable vaccines, smallpox and measles, show benchmark responses that says that it is important for B cell memory development with the assistance of T cells. So this brings up very important questions. How much emphasis is needed in vaccine strategies for T cells, memory B cells, and conserved viral regions? Do we need to consider that as well? Can memory B and T cells, if we do develop them, protect against the setting of an acute viral disease with a short incubation period? You want the antibody to be there early on. So that's an important consideration. So from the perspective then of how important T cell immunity is, recent publications show that a robust T cell immunity is important in convalescent individuals with asymptomatic or mild COVID infections. And that comes from a series of recent studies, 2 of which I will review here. The importance of memory T cell responses as a persistence of immunity. First is the Swedish cohort study from Dr. Sakin et al., published in Cell in August, where the presence of COVID-2 specific CD4 and CD8 T cell responses during the acute phase infection correlates with the presence of highly cytotoxic T cell phenotypes in the blood. In the convalescent phase, the discovery that polyfunctional and durable display of CCR7 memory T cells are important. Remember that CCR7, I'll come back to that. Durable and functional memory T cells were also detectable in convalescent subjects with asymptomatic or mild infections, and even in exposed seronegative family members, people who thought who didn't know that they were even infected. The memory T cell responses developed against nucleocapsid membrane as well as S protein. Similar findings were recently also provided by Lebert et al. So how do we cater for B and T cell responses? Well, the nucleic acid vaccines may already do it. That'll be fantastic. But I'm also now going to introduce you to the peptide-based vaccines and the viral-like particles, which can deliver minimal structures, parts of CoV-2, such as the intact S protein, S1, S2 subunits, RDB, or peptides. Examples, vaccines from Novavax, Sanofi, Johnson & Johnson are examples. This involves either the use of multiple particle types, mentioned them earlier, liposomes, polymer, lipid nanoparticles, or a new generation of viral-like particles which are constructed by assembly of mammalian, insect, or plant virus proteins that self-assemble and can then deliver the vaccine subunits. An example is iBio tobacco plant virus particles, which are comprised of self-assembly proteins that is produced by fast farming technology. The viral-like particles are potent, but as the downside may raise antibodies that cross-react with other viruses. All of the above formulations are delivered with non-nucleic adjuvants, and then also have one additional advantage that you can add targeting ligands to take it to specific parts of the immune system, such as the lymph node. I'll give you one example later on. So with this and the understanding that T cells are also important, we see widespread use of epitope mapping and informatic technology to predict peptide sequences that potentially can be used for vaccination with B and T cell epitopes. While most of these efforts are still out there, don't have a lot of data, the preliminary results are quite important. For instance, publications by Dr. Grufoni, Ahmed, and Gautier mapped multiple B and T cell epitopes to CoV-2 for S1 nucleoprotein and conserved viral domains. They're often shared with CoV-1. The B cell epitopes, very advantageous because you can give them to anybody. They don't require HLA presentation. More problematic for the T cell epitopes, which requires HLA presentation. But the studies from Dr. Ahmed and Dr. Gautier have shown that you can actually design a number of peptides that bind to multiple classes of HLA. For instance, in Dr. Gautier's study, the prediction of 4 different peptides that can be presented and protect potentially 100% of people. The advantages of epitope-based vaccine development is that it's inexpensive, scalable, safe, that you can have humoral and T-cell-based immunization, you can combine them, and that there is also ongoing informatic-based efforts, as shown here on the web-based epitope mapping domains, where informatic-based updates provide the potential to do iterative vaccine design if there are any changes in viral antigenicity. I'm going to show you one example from a vaccine nanoparticle that we developed in CNSI by making use of a polymer backbone in which we can deliver subunits or epitopes. We've added a very strong Th1 adjuvant. We can add targeting motifs, and where the particles after the first 2 injections effectively raised IgM, that's a primary immune antibody response at day 21, in which the adjuvant had a much stronger effect. At this moment in time, the IgG antibodies were still negative, but with a 3rd vaccination and subsequent bleeds at day 35, 42, and 49, we have a sustained high production of IgG antibody titers. I do not know to what extent these vaccines are going to play a role, but if I look at what's happening in the nucleic acid vaccines, I can also see that the epitope mapping may result in mini-gene nucleic acid platforms for multivalent constructs that can generate B and T cell immunity in scalable nanoparticles. And what I'm talking about is that instead of using peptides, you can use string of bead vaccines, which is based on finding epitopes, for instance, for an infectious agent, as well the epitope sequence, as well as flanking sequences to allow you to make a mini gene in which there's deliberate spacers put in. This is quite a strenuous design process, but ultimately leading to mini genes which, if correctly cleaved, delivers at the same time multiple epitopes to the immune system. In a recent paper by Dr. Yomarkovich, they constructed 65 33-mer peptides with sequences to diverse B and T cell epitopes. They used it in tandem with Th1 adjuvants, and the epitope selection that took place was against conserved viral region as well as more variable regions, such as the S region. The selection for diverse HLA antigen presentation to stimulate CD4 anti-CD8 T cell responses by MHC1 or MHC2 presentation, the selection of linear and conformational B cell epitopes, and the selections made with the highest degree of dissimilarity of the viral sequences to the human immunopeptidome to enhance the immunogenicity. String of beads vaccines have experimentally been shown to be successful for cytomegalovirus or influenza. And I wonder if we will then also see, based on this report, the emergence of string of bead vaccines in the future for SARS-2 if the other vaccine types should not work out. Also want to say something about the vaccination efforts that have been learned recently in a paper that still has to come out in Cell next month. The importance of the lymph node structure in durable immunity that requires B cell and T cell cooperation in COVID-19. And this is from a paper by Dr. Parodi et al., where he describes that in acute COVID-2 patients, there is a marked reduction in lymph node structures known as germinal centers. You see here a lymph node, and the areas here are germinal centers. And these germinal centers contain a class of B cells that switch to antibody production that is high affinity and confers memory. Ultimately, these B cells also get into the blood circulation. And in the development of this interaction in the germinal center, important cooperation from a CCR7 memory B cell that evolved into what is known as a THF, a follicular helper T cell. And then importantly, the discovery that in COVID-2 patients, lymph node structure is disrupted. They don't make good germinal centers. They do not have much follicular helper T cells around. And the recognition in the vaccine world of the critical need, if you want to develop good vaccines, is the development of platforms and adjuvants that target the follicular helper T cells to the germinal center. So it's interesting that in making a RNA lipid nanoparticle for vaccination in Zika, influenza, and HIV vaccines, Dr. Parodi et al. Have recently shown that the lipid-based nanoparticles do provide potent THF and germinal center cooperation. And the results were also confirmed by Dr. Lindgren with influenza virus in rhesus monkeys. So in the lymph node structure, the cooperation between the follicular helper cell and the B cell is important. Another cell type to present antigen that's important there is also known as the follicular dendritic cells. So the FDCs are important. And why I'm showing you this is that in the world of nanotechnology, an important discovery from Darrell Irvine's laboratory is the role of glycosylated proteins in order to get to the germinal centers. And in vaccine development against HIV GP120, if the protein itself is delivered, it only collects in the subcellular sinus in the lymph node, but it doesn't get into these germinal centers. If the HIV 20 is folded into polymerizing nanoparticles that are rich in glycans. The glycans drive a very high expression of follicular dendritic cells, T cells, B cells in the germinal center. And the secret ingredient here appears to be the glycan, which binds a certain complement binding protein. It's also known as mannose binding lectin. And it's the complement activation in this vaccine that takes the vaccine to the germinal center and makes effective antibody responses. Darrell's lab has now also done it for other HIV proteins as well as for influenza proteins. So the homology of the COVID-S protein with related class 1 fusion proteins in influenza and HIV is probably going to play a very important future role for vaccine development by consideration of the pre-fusion conformation through mutagenesis, as well as the glycosylation status of all of these proteins. And here I show just for the COVID-2 that all the major epitopes are heavily glycosylated. Finally, in the vaccine part, I'm going to say just that an important consideration is the duration of the immune response and the question of reinfections. COVID-2 has good genetic proofreading with low sequence diversity, and there's actually little indication that reinfection is going to play a big role. However, for COVID-1, it has been shown that a mutation in the RDB domain can provide escape from neutralizing antibodies. It's also interesting from a paper published by Dr. Betty Korber recently that for COVID-2, an S-domain mutation happened as the virus migrated in the orange here from Wuhan ultimately to Europe. Within 1 month, an S-domain mutation took place at a cleavage site for furin, which is a possibility, therefore, that it can have an influence on the RDB presentations. Patients infected with the mutagenic viral form shed more viral RNA compared to the wild type with significant higher antibody titers. And so the possibility that pandemic persistence could lead to accumulation of immunogenically relevant mutations in the populations as vaccines are introduced. And Dr. Korber published a very nice online accessible designed bioinformatic tools to act as an early warning strategy to evaluate spike protein evolution during the pandemic. My last 3 slides is going to then briefly just say that in the world of vaccine, the lipid nanoparticle also has been used in other attempts at vaccination for cancer, for instance, where these lipid nanoparticles deliver nucleic acids to a cancer environment to assist immunotherapy. Of course, cancer, with the death of a cancer cell and shedding of cancer antigens, can invoke the development of immunity in the regional lymph node where it instructs T cells, and these T cells come back to the cancer. Our own work in this regard focuses on what we call an endogenous vaccination strategy for cancer, in which we deliver nanoparticles that kill cancer cells specifically to kill them in a way that has been described as immunogenic cell death, where the cancer cell dies by expressing calreticulin on the cell surface as an eat me signal for the immune system, releases 2 adjuvants, HMGB1 and ATP, to drive the immune function. And this technology is being introduced on the background of another major development in cancer, namely checkpoint blocking antibodies. I'm not going to go into details here, but checkpoint blocking antibodies have made a major advance for certain cancer types, where compared to the uniform mortality of the cancer, the immunotherapy can provide up to 20% of people with a response that can include a cure from the cancer. And why the other 80% don't respond is thought to involve whether the cancer is loaded with cytotoxic T cells to be able to respond to the antibody. So you can have a cold immune environment, the hot immune environment will respond, but if you consider that the other 80% of people that do not respond, perhaps you can wake up the immune system by providing of immunogenic cell death. And we do so by delivery of chemotherapeutic agents that kill the cancer cell anyway, but have immunogenic effects. And I'll show one example here, and that is a nanoparticle that developed that is analogous to a liposome, except this one has a lipid bilayer that covers up a mesoporous silica nanoparticle in which we can do high drug loading with chemo agents. One chemo agent that can be loaded quite effectively is irinotecan. If you test this nanoparticle out in a very rigorous immune-resistant cancer, namely pancreas cancer, the primary cancer and its metastases respond very little to free irinotecan. If you introduce an irinotecan liposome, you can see the primary tumor somewhat reduced, but metastases still there. Our irinotecan nanoparticle, however, wipes out all the metastases, and there's very little primary tumor available. In a recent important discovery, the irinotecan noted to also induce immunogenic cell death responses, and that the cancer also induces one of the checkpoint blocking receptors, PD-L1. So I'm not going to go into all the details, but in addition to the chemotherapy response that I've shown in the previous slide, the introduction now of the silicosome with an anti-PD-1 antibody dramatically prolongs, and we even have survival animals in the group. So I'm going to acknowledge, of course, for work like this, it requires hard work by a team. Our associate professors, Wan Meng, Chan Jia, our postdoctoral fellows, Jiansheng Lu, Casey May, Sean Allen, Chu Li. Chu Liu is the person also who made the COVID vaccine, our staff scientists and students, and then also important from an institute perspective, our combination of nanomedicine, precision antibiotics, environmental health and safety, CMOS, renewable energy research, all driven with a philosophy of basic discovery for translational science and commercialization, to which CNSI has very successfully developed an entrepreneurial platform where 35 companies incubate, and also a proposal facilitation unit where we have groups work together and are able to generate funding levels that is way above the current funding levels. Thank you very much. Be happy to take questions.
Thank you very much, André. And if you stop sharing your screen, I'll bring up the questions.
Are you? Is the screen okay for you, Paul?
There we go. Yes. I think
yeah.
We're,
okay.
Okay. Well, let's see. Our first question is, Professor Nel, thanks for your wonderful talk. The vaccines are very important. SARS broke out in 2003, 17 years ago. Why is there still no vaccine for it?
Well, that's a very interesting question, because I showed you that for COVID-1, they tried to make a vaccine to the S1 and the RDB, as well as inactivated virus, and succeeded to show that you can get protective vaccination. However, two things happened. The one is that when those animals ultimately were studied with viral infection, the side effects developed that I showed you earlier on. And second, that the COVID-1 viral epidemic was much shorter lived and went away, so that the necessity to continue with a vaccine unfortunately was called off. But I can just tell you that a lot of the lessons that was learned in such a way that when COVID-2 emerged, a rapid response from a number of entrepreneurs around the world have enabled us to use what knowledge was generated to make the current set of vaccines.
Interesting. Second question is, as you said, we've changed how we approach vaccines. What have we learned from these studies that will carry forward? It's actually a nice continuation from these studies to the next dangerous viruses that we face.
Well, I think that's also an excellent question, because what I've tried to show you here and why we've went ahead in CNSI and making our, one short attempt while we already know there's 140 other vaccines available. I think the importance here is the ability to quickly translate what is important about a pathogen against scalable technologies that can be rapidly brought to bear is what will happen in future vaccine efforts. And so it's going to have a huge, huge impact. And I showed you that it's going to have a huge impact, I don't think only on infectious disease for COVID, but on many other infectious disease where we deal with, for instance, the equivalent of a spike protein that is also plays a role for HIV, plays a role in influenza, respiratory syncytial virus, etc. And then the other important recognition is that you can work with nucleic acids instead of having to do protein purification or technologies where you require large-scale production of cells in order to drive your vaccination. Another aspect I've indicated is the smart design of adjuvants for your vaccines in order to skew the immune response so that you get protective rather than interfering immune responses.
The third question, for this important vaccine research, is there a platform for scientists to share information and or data, or is everyone working independently?
Well, so I have to be careful in answering that question, because I'm not one of the people currently working with any platform that is trying to market the vaccine. I would say that publicly available information has played a key role in research that was published on the early COVID events. Once that gene sequence for the COVID virus became available, it inspired certain companies to be able to do prediction making for making their antigens within a matter of days. So I would say public information has played an important role. There's currently also online, I've told you about the published online T and B cell epitopes from, I showed you 3 scientists who have released elaborate information about T and B cell epitopes. I've also told you about the studies from Dr. Betty Korber, in which they're tracking an informatic system about any mutations that is developing in what is currently the key driver for protective immunity, namely the S protein. So I personally think that the more information that is shared between scientists worldwide, the more advantageous it's going to be in order to effectively combine these technologies for the betterment of mankind.
Thank you. The next question is, thanks for your great talk, Dr. Nel. Advance's main research labs generally take a while to be translated into the clinic, especially because of the very long FDA approval process. Can you shed some light on what can be and is being done to expedite this process for COVID-related research?
Well, again, I don't want to be talking on behalf of federal agencies and developers, but I can just say that the current rate at which new vaccines are introduced in phase 1, phase 2, phase 3 clinical studies and is being tested out is absolutely a new era and a new domain in which we've entered. I think it is also very important that those studies be done responsibly and carried out, as we have seen recently from a severe side effect. It is important that the phase 3 clinical trials be completed in addition to the phase 1 and 2. And but I have very good reason to believe that if the evidence is demonstrated that an agency like the FDA will act early on. But I also think it's important that no action is taken until it is proven that the data that comes in shows that the vaccine is effective and safe.
Very good. And then penultimate question here. COVID-19 is varying, mutating in many different places already. Which approach is likely to be better with these variations, DNA or RNA or peptide vaccines? So it's really with some.
Wow, so that's it's an interesting question to which I don't have the answer. Let me just say that although COVID-19 is mutating, I told you that the virus have a genetic proofreading capability that provides a lower rate of mutations than what we see in the corona cold viruses. So from that perspective, mutagenesis is not as fast. There's also very little data at the moment about reinfections, but there are 1 or 2 reports where that have happened. I would say that if in the current vaccination attempts we get 1 or more vaccines that are effective, that those vaccines will, in terms of the necessity, have to be developed and manufactured in large enough quantity to be used in the current COVID crisis. If any of the vaccines fail to develop long-term immunity, and if we do see that immune escape is an important problem, then I think we will enter a domain where further vaccination efforts will be important. And in that regard, the peptides may be very important to make sure that you get a correct blend of not only neutralizing antibodies, but also T cells. I've also shown you that for both DNA and RNA, the technology now exists to make vaccines, mini gene vaccines that can express multiple peptides. Those aren't developed to full extent yet. What is better, DNA or RNA? I think they both are going to turn out to be very good. At the moment, I showed you that for the RNA, there are some advantages of why the technology may be more rapid to implement. But we still need to learn a lot about all the questions you're posing now.
And then, then the last question from the audience. Dear Professor Nel, fabulous. As you know, China is a very strong group led by Professor Wei Chen. I believe she's at the Beijing Institute of Biotechnology, and just announced very positive results of their vaccine in human trials. Can you comment on this result, and might it be a resolution for COVID-19?
Unfortunately, I cannot comment much on it at the moment, but we'll be looking out at all the different publications that come out. And, perhaps if Professor Chen contacts me once I've had opportunities to evaluate, I'll be happy to give a response.
Mm-hmm. Well, thank you for this wonderful talk and the great interest it's generated. As I mentioned at the end of Paula's talk, right now we'd normally be seeing each other in person and walking across the stage. We're just across town in West Los Angeles, talking to each other through Beijing. And so let me thank you on behalf of Professor Alice Zhang and myself for connecting the world together and sharing, this incredible insight into the progress in vaccines and nanotechnologies contributions to it. This is just such an insightful and thoughtful talk. I really appreciate it, and I speak on behalf of the hundreds of thousands of people who are watching online and will watch later in the recording that'll be posted on YouTube. So thank you very much for this terrific.
Well, thank you for the opportunity to participate, and I wish all of you the very best and keep safe amidst the difficult era that we're going through.
Indeed, yes. Thank you. And now